जानकारी नहीं है माइक्रोबायोलॉजी का मेरा मेरे पास में माइक्रोबायोलॉजी है वो नाम गलत लिखे हुए हैं। हाँ जी करवाना हाँ जी करवाना हाँ जी आपने जिसके ऊपर ना लिंक भी लिखा है वही ली है और नीचे माइक्रो हाँ जी माइक्रोबायोलॉजी देखिएगा मैं अभी आ जाता हूँ। हाँ जी 5 ठीक है ठीक है ठीक है ठीक है
join our session. Hello.
Yes, sir. Hello.
Good
afternoon, sir.
You are not visible.
आपको जो भी ऐड टू स्टेज करना पड़ेगा जो भी बोलना है से
Hello. Sir, the presentation I cannot share over it. Yeah. Hello. Is it audible, sir?
अभी अभी बोलिए
Tamnoy, you are not visible. Your presentation is visible to us, but you are out of the scene. Chalo, it's okay. You just go ahead.
Okay, sir. Sir, good afternoon, everyone present here. Sir, I am going to present a topic on the overview of smart drug delivery system, its technology, advancement, clinical challenges, and future perspectives. So, the smart drug delivery system, as we know that in pharmacology, is the main work that the pharmacology goal is to deliver the drug at right time with the right dose at perfectly particular definite sites to give the appropriate effectiveness. So here the conventional drugs that which we are using nowadays sorts fall in this ideal. So the traditional forms of dosage forms have now emerged with this new drug that is the smart drug delivery system. And the smart drug delivery systems are often referred to as the advanced technologies that helps in transporting the therapeutic effects to the particular targeted organs or the particular targeted sites. These are oftenly responded or referred to as the stimuli responsive or intelligence or demand and delivery systems. They are normally classified at different sites. And for this smart drug delivery systems, there are different types of regulatory perspectives which are needed to be followed, or some of which are being followed by as per the regulatory guidelines of US FDA and EMA, which oftenly categorize them as smart technologies for the drug delivery systems. Some of them have the combination products that FDA classifies as SDDS or of combination products like drug device or biological devices. Some are evaluated as combination products. Then we have the nano medicine guidelines, which the FDA have approved, still not fully, but some of them are in the clinical stages for the new developments of the medicines. Then the EMA, that the European Medicine Agencies, is they also complex the centralized process of SDDS. And in the India, the process has been regulated by the help of CDSCO, or Central Drug Standard Organizations, which classifies and maintains the new drug classifications. and the new drug research and development work along with the nano pharmaceutical guidelines. Next, we have the classifications of SDS, that is the smart drug delivery system. There are different classifications for smart drug delivery systems, some of which are and mainly works with the stimuli responsive systems. For the stimuli responsive systems, there are two types, that is the endogenous or the internal stimuli response system and the exogenous or external stimuli response system. In the internal stimuli response, they rely on mainly basically on the biochemical or physiological variations with the body microenvironments. For example, it's reference with the pH responsive systems where it capitalized with the acid nature of the pH is to give the result. For example, the polymericyl acid, the PMMA nanomolecules or the nanocarriers or the liposomes that are polymeted with the pH sensitive, if like the dopamine, to give the acidic environment for the platelet release. Next, with the deoxyribose system, which is highly concentrated on the reducing agent, and for example, the disulfide crosslinks of the nanogels and the enzyme responsive, for example, the peptide conjugated liposomes. Then the exogenous or the external stimuli responsive systems, this mainly applies on the external or clinical control physical that triggers the initiate drug release. For example, we have in this the magnetic responsive systems, which contains the example of super permanent iodide oxide nanoparticles, which encapsulate with the hydrogels or the liposomes. Next is the lightweight responsive systems. And for example, we use here the gold nanobars with the UV light. Then we have the multiple responsive system or the dual responsive systems, and where the limitations and the targets are the multiple response give more than one stimulus. For example, pH and the temperature dual response systems, nanoparticles, which helps in the triggerness of this is examples. And the self-regulated or closed loop systems, these are the autonomous or the responsive that are specified biomarkers that release the biomarker concentration, normalizing the hormonic or the effect of the drugs. For example, the glucose responsive systems for the diabetics is where the PBA based hydrogels or the microneedle patches containing the glucose dioxide is helps in the release, is when the increase in blood glucose level, they help in the enzymatic reaction to bind and release the insulin, and when that it gets the normal base of the insulin level or the diabetic level drops, then it stops the prevention or the movement of this is insulin to the body to maintain and the insulin or the sugar level of the body, so that it does not cross or goes down to hypoglycemia. Then the emerging technologies is that we are facing with these new modern then drug delivery systems or the smart drug delivery systems. These are the nanoparticles. These are produce various carriers and helps in biomolecules particles with the help of liposomes, nanomolecules, lyosomes, and silica or metal or different type of metallic nanoparticles. This helps in the response to different type of internal and external stimulus to control the release of the medications. They link with the different ligands and the receptors to give the target achieved and to give the target oriented drug therapies to the organs or to the sites. Next, with the device and wearable, there are different types of smart drug delivery systems that are associated with smart wearable and smart devices. As that comes under implemented drug delivery systems that are implemented in the body. They are implemented with the infusion pumps, which help in the painkiller or insulin release rate, with patches, which helps in the diffusion systems with the skin technologies, which diffuses through the skin in for the cuts and different types of infections. Next, the artificial intelligence or the AI systems that are emerging nowadays in different fields is along in the field of medications. and they help in optimizing the dosing and different types of treatment with the network delivery systems and the devices.
Tanmoy,
Yes.
yes, sir, your time is about to finish, so try to make the just sort up,
Just compile it.
compile it.
Okay, sir. And the key challenges that we are facing nowadays is in this new drug delivery system are the biocompatibility with the carrier, because there are different researches that are going on and not as we are going to do individual target specific. So biocompatibility is a main problem that's challenging us with the reaction and toxicity. Then the nanomaterials for which we are building the nano particles from which there is also a specific here. Then the manufacturing and complexity, maintaining the quality, because maintaining the quality of this smart delivery systems are main challenging to us. Then the economic valuability and the patent and the patient adherence with the infrastructures for these delivery systems. Next, for the future trends and the strategies which are being used in the smart drug delivery system helps in removing the passive environmental through different autonomous and biohybrid and digital integrated platforms with the help of precision medicines. With the help of the smart delivery systems, we will finally have the precision medicines and biomarkers which help us to get low or adverse effect. Then we have the sustainable materials that focus on the development and biodegradables of the nanomaterials with complete safety and efficacy. Smart packaging, which helps control the cost effectiveness. And in the conclusion, and if I conclude my saying with this,
अब इसमें से करो उसको ऐड करो.
yes, I want to conclude this presentation with that the smart drug delivery systems are nowadays replacing the fundamental and the traditional medicine ways, is, and it helps in different and overcomes with the different procedures and new future perspectives with the help of the safe to dynamic intelligence, where the SGS translates the passive encapsulation to active if the calculations and then represent the front line of the precise medicines is with digital technologies and AI helps widespread the clinical absorptions which require significant barriers, helps in manufacturing of and representing and getting solutions with new drugs, which helps in development of the new treatments and which are now helpful in achieving new treatments which are complex, yes, and challenging to the research organizations.
Add to the stage button.
Hello,
Yes, sir.
hello. Saha, what was the key area on which you are looking forward after this particular presentation?
So I am just looking forward as nowadays there are different types of mainly medicines are lagging in the field of cancer treatments, and as in cancer treatments we mainly use the radiation treatment for various types of cancers, which affect the different healthy cells near about the main affected cells. So in the help of the nanotechnologies, there are options that we can just or specified on the target oriented and just keep the healthy cells as it is.
Okay. So one more question, sir. Can we make the inclusion of all this technology in the present medical science as we are treating the diabetic patient?
Yes, sir. We can implement, and there are different researchers is already going on with this implementation of the smart drug delivery system, because it will be more effective to the patients which are having blood sugar levels, because it automatically will release the insulin in the body when there will be high blood glucose level in the blood, and automatically it will reduce the amount of the blood sugar in the body, and it normalizes and stop the effect when it reaches the normal level.
Okay. Saha, one more question. While we're talking about the Indian continental, we have to look after the pocket of the customer.
Yes, sir.
So, what about the cost?
10 से ज़्यादा है.
Cost
Remove करो.
effective.
So this is the main challenging, because the cost is getting high due to the materials uses. So there are been different materials and different technologies using in it, so that we can reduce it the cost, and it could affect and it could be helpful and fit in the budgets of normal general publics.
Okay. Give me any example which is quite present existing smart delivery drug in Indian medicine market. So give me some example, some instance.
Means you are asking a question that any drugs that is present in the Indian
And
market for the
yes,
existence of this smart drug.
I could. And yes, any pharma company which is being working on this area for this as smart drug delivery system as DDS. And you have any update for any medicines which is being present available in the Indian market when we are talking about the smart drug delivery system? Any medicine?
Sir, there was nanotechnologies that have been used in the gold nanotechnology have been in the stages of clinical trials for the delivery of systems of cancer systems, which help in the radiation science and contraction of the cancer cells. Yes, and some of them are working on it, and there are some certain polymers and DNA means some technologies that are being used with the gene therapies is with these technologies for the cancer treatments, which helps in the mutations mutations of this specific site of the muscles or the cells.
ठीक है. Thank you, Tamno. And yes, so nice presentation. But yes, there's a lot of scope of improvement. Thank you. Thank
Yes,
you.
sir. Thank you, sir.
Thank you.
इसे नेक्स्ट कर लो, मेरे रघु है इसमें, इससे आगे मैं
नीचे लेगा स्क्रॉल
मेरे रघु है इसमें,
लोगों भी लेगा
नहीं होगा ऊपर, वो रहा देखो, ये दिख रहा है ना, इससे सेल जैसे ऊपर ये रहा, इसको ऐड करो,
ये
इसको ऐड स्ट्रेच कर दो पहले इसको हटा
इसको
ऊपर वाले
इसको
अरे वो ये तो मनीष सबको ही आएगी,
ठीक है इनको बोलो अभी अनाउंसमेंट करके बोलो इनको शेयर करेंगे अपने
मेरे रघु को बोलो।
yes, Rahul Raj, you just share your PPT.
Hi, sir. Hello.
Yes. Please share your PPT also on the side of your screen.
And you have
Sure,
time
yeah.
limit 8 minutes.
Yes, and you have the time limitation of the 8 minutes. So try to
Sure,
make the best presentation in that particular time.
Sure.
Okay, you just started.
It's taking time to. Can you see my screen, sir, or still processing?
Yes, I can only see you.
Yeah, sir. I mean, I don't know. I just uploaded the PPT. It's taking time. Yeah. Can you see the screen now?
चलो सर डाउन सेंड
Or is it not visible, just me?
you add to stage
Yeah.
Yes. So,
Hope you can see
yes.
my screen, sir. Yeah. So, yeah. Good afternoon, everyone. Today I'll be presenting a study titled PD1 Blockade in Mismatch Repair Deficient. Particularly, this is categorized in local advanced rectal cancer, and this research was conducted by Sarek and published in New England Journal of Medicine in 2022. So, moving into the background of it. Just a second, sir. I don't know why it is not showing. Let me present my screen again. Share screen. Yeah. Yeah. Hope you can see again. Hello.
Nothing is visible on the screen.
Just only the background.
Only background is written on your screen and nothing is accepted.
Still, you can't see, sir. I just started sharing my screen.
Yes, we can't see right now.
Let me stop sharing and reshare it again. Just a second. Is it visible now, sir?
Yes, yes, it is visible.
Yeah, thank you. Uh, so the background is rectal cancer. This is the most commonly treated. I mean, the rectal cancer is obviously the most highly morbid condition in the entire human history. So when it comes to the treatment of it, rectal cancer is usually treated with a combination of chemo radiation, which is then followed by a total mesorectal excision, which is exclusively performed by a surgery. And when it comes to the rectal cancer, 5 to 10% of it is
Sign है वहाँ पे
sorry.
ना
Okay.
आपको
Sorry, ma'am. Can you hear me?
Yes, carry on. You just carry on.
Yeah. Sorry. So 5 to 10% of it is a subset of rectal cancer, which is exclusively characterized by the deficient mismatch repair, which is a DNA mutation within the cancer cell. And because of this, 5 to 10% is accountable for the advanced rectal cancer. So when it comes to, let me share the next slide. Aims and objectives of this particular study. The aim of this study is to understand or study how neoadjuvant neoadjuvant treatment with PD-1 inhibitor, which is dostarlimab, could eliminate the need for chemotherapy as well as surgery, which is the combination which is exclusively used as a treatment option for rectal cancers. When we see the primary objective of this particular study was to determine what's the portion of the patients who achieve complete clinical response within 6 months period of treatment. And the objective will be assessed using MRI imaging, endoscopy, as well as digital rectal examinations. When it comes to the second objective, the second objective is to evaluate the safety of the drug and to understand what's the pathologic clinical response, and to also understand the sustained treatment overall duration. And what are the methods used for this particular study? Is the eligible criteria is limited to only stage 2 and stage 3 rectal cancer patients who are diagnosed specifically with, again, uh, the DMMR status, which is just a second, which is deficient mismatch repair, the change in the DNA. And the patients, there is also another criteria where the patients should not undergo any prior treatment. So in these patients, they receive the drug dostarlimab at 500 mg intravenously every 3 weeks for 9 cycles, which is approximately 6 months of therapy. And when we see the assessments, the baseline will be assessed using MRI pelvis and PET CT scan, and it also be evaluated using endoscopy with biopsy. The clinical examinations were performed for every 8 weeks, and after 6 months, which is the course duration, patients again underwent into MRI, endoscopy, and biopsy to assess what is the complete clinical response of that particular patient. So when it comes to the complete clinical response, this is usually defined as no detectable tumor over the period of time or once the course has been completed. And this can be detected through, again, the above mentioned baseline parameters like MRI pelvis or CT scan or endoscopy or biopsy. And if the patient has not achieved the complete clinical response post the duration of the treatment, then he has to eventually go with the conventional therapy, which is the chemo radiation and the surgery procedure. Moving into the results, while the interim analysis has been pulled in the first 12 patients, it was clearly noted that there was 100% clinical response towards the drug in 12 patients, and also there was no evidence of tumor progression, which is the growth of tumor further. And also there is an important factor that the adverse events has not crossed grade more than grade 3 or grade 3 and beyond. So this is another milestone which was achieved in the results. And when it comes to the median follow-up, even after 6.9 months, the duration of the treatment, there is a complete clinical response throughout the period. And this is just the clinical response rates which were received during the treatment course for those 12 patients. And moving into the safety profile, as mentioned, like only grade 1 or 2 adverse events were reported, and most commonly reported were dermatological reactions or fatigue, and 50% of the population have experienced nausea. And among the patients, there was no delay or discontinuation of the treatment, and the safety profile is more or less comparable or better than the regular conventional therapy. The discussion and implications. So when it comes to discussions, the study demonstrated an unprecedented 100% clinical response to the drug, and it also excludes one major. It also has one major advantage, which is potentially preserving the organ uh, by avoiding the surgery. So these two are one of the key challenges, but however, there are a few limitations, like the sample size is little less, and also long-term data is exclusively needed to understand how the drug works. And moving into the conclusion, as mentioned, that the neoadjuvant PD-1 blockade with
Yes,
dostarlimab, yeah,
Raz, thank you for nice presentation.
yeah,
So move to the slide 5, fifth
just a second,
slide.
just a second, sir. Yeah,
Fifth slide. So here in methods of assessment, you took the complete clinical trial, no residual tumors.
yeah,
So in this, you told me in the result that is there is 100%.
yeah.
Yeah. So would you make some point on that non-CCR chemo radiation plus TME?
Uh,
In
CCSR
the
and sorry, sir.
non-CCR,
Okay, okay.
and that is a chemo radiation plus TME. So
Yeah, yeah, yeah.
what was your intervention? You told the implication of this particular medicines.
Yes, sir. So dorzotalimab is basically a PD-1 inhibitor where there is a PD. There is a receptor in the T cell which the drug gets bind so that the tumor cell does not bind with the T cell and does not give the necessary immune response to reduce. To reduce the immune response while fighting back with the tumor cells. So this is the main objective of dostarlimab. But when we see the methods and other things, you asked me like how it is achieved 100% clinical responses. Again, the disease itself is 5 to 10% characterized of DNA mutation in the rectal cancer. So this is one advantage, that the drug is directly binding with the T cell instead of the cancer cell being bind to the T cell, so that the achievement of the drug response is 100%.
So you want to say your intervention completely work on the T cell, and there is
Yes.
no involvement of the B cell or other immune factors.
Yes, yes, sir. So
Yes.
basically, the drug binds with the T cell, therefore, it stops the cancer cell to bind it, and therefore keeps the immune system active so that it detects the cancer cells and kill them or defeat them accordingly.
Oh, okay. Thank you, Raz, and yes, and good presentation. Keep it up.
Yeah.
Thank you.
Thank you, sir. Thank you.
इसको ले लो वाला इससे नहीं अप साइड में है, इससे ऊपर हाँ इनवर्ट नहीं किसको से बताऊँ फाइल में अनु अनु है कि अनु नहीं है, मनीषा मनीषा है,
Manisha.
मनीषा
Add it.
इसको ऐड बीच में क्लिक करो, इसके ऐड हो जाएगा। हाँ, ऐड टू द स्टेज।
Manisha.
Hello, hello, sir.
Uh,
Yes, sir.
hello. Yes, please share your screen.
Okay, sir. Just a sec. Share screen. So, second, second. So my screen is visible.
And
इसको देखो, आप जाके देखो
नीचे scroll.
नीचे scroll
Yes.
Yes, sir. My screen is visible.
Yes.
Can I start?
Not yet. Yes. Not yet. Not
Okay,
yet.
sir. Not yet.
जोड़ दिया
Sir, अभी my
अभी
screen
भी
is
नहीं,
visible or not?
नहीं अभी नहीं है जो, not yet.
Just a sec, sir.
ये है, फिर अनु को फोन कर लेते हैं और फिर मनीषा की बात लगा लेते हैं।
एक second. एक second. Sir, my screen is visible.
Not yet.
मनीषा,
Manish.
आप ऐसे स्टार्ट।
Yes, right now it's so okay. You just
Visible?
started. Yes.
Okay. Okay. Good afternoon, all of you. I am Manisha. Today I am presenting my research on. Just to wait a second. My research on demographic profile and bacterial spectrum of respiratory tract infection. This is a laboratory-based culture-positive isolate. And next is the first of all introduction.
Manisha,
Sorry?
Manisha, just move to the main part of your presentation. You just move to the main part of your presentation.
Sir, this is the aim.
You just go
Sir,
on the aim, then results and discussion.
Yes.
Okay, ma'am. Okay. So my research aim is to analyze demographic profile and bacterial spectrum of culture positive respiratory infection. So
Manisha?
इसके अंदर जो है मैंने yes, ma'am.
Manisha, scroll down your slide also. Scroll down your slides also.
चेंज करो ना स्लाइड्स को भी।
मैम
आपकी एक slide-
चेंज ही है।
नहीं, आपका
हमें-
सिर्फ फर्स्ट स्लाइड विजिबल है।
हाँ, जिसमें आपका डिटेल
फिर जहाँ पे ऑब्जेक्टिव है उस स्लाइड पे क्लिक करिए आप।
ऑब्जेक्टिव पे क्लिक कर रखा है मैम मैंने। शो क्यों
कोई
नहीं
बात नहीं,
हो
आप
रहा
बता
है?
दीजिए, कोई बात नहीं।
चलो, go ahead.
Okay. So to analyze the age-wise and gender-wise distribution of culture-positive respiratory isolates, to identify the most common bacterial pathogen causing respiratory tract infection, to assess the antimicrobial susceptibility pattern of the isolated bacteria using Vitek to compact machine system, to evaluate the occurrence of multi-drug resistant organism among respiratory isolate, and to generate local laboratory-based data that may support empirical therapy, antimicrobial stewardship, and infection control measures. So this is the materials and method that I use in this research. Out of 46 sample, 460 samples I used in this research, and 191 were positive. I get next. Next is these are the bacterial isolate, different bacterial isolate that showed that I got in my research. And next is the result. So a total number of 191 culture positive respiratory isolates. Out of them, 115 isolates were found as a male, and rest 76 isolates were found as a female. So which means that the infection were more widespread among males. So This graph shows the male candidate who's the most infection. Next is the age-wise distribution. This graph shows the highest age group, highest bacterial infection was in more than ages like 61 to 80 years age group patient acquires more higher infection, and 41 to 60 years age group moderate infection, and less infection was in a 0 to 20 years. So conclusion is that ki elderly population was most affected. Next is the bacterial spectrum, predominance of gram negative बैक्टीरिया. मुझे इस रिसर्च के अंदर सबसे ज्यादा जो है वो ग्राम नेगेटिव बैक्टीरिया ही पाए गए हैं. Like this is Pseudomonas aeruginosa is found the most, number is 59, and less is the Streptococcus pneumoniae. So next is this table show the age-wise distribution of different bacterial isolates. So इसमें इसके अंदर ने टेबल बनाई है. टेबल के अंदर सबसे ज़्यादा हाईएस्ट जो बैक्टीरियल आइसोलेट है वो पाए गए हैं मोर देन 60 ईयर एज ग्रुप में. लाइक जैसे इकोलाई वो मोर देन 60 ईयर्स में 60.42% पाया गया है, एंड सुडोमोनास इज द 48.33%. सबसे लेस जो है वो है ग्राम पॉजिटिव के केस में मुझे मिला है मोर देन 60 ईयर्स केस में. So, ठीक है, तो इसके अंदर सबसे ज्यादा आइसोलेट जो है वो ग्राम नेगेटिव के पाए गए जो कि मोर देन 60 ईयर्स के एज ग्रुप में पाए गए. नेक्स्ट इज द एंटी माइक्रोबियल रेसिस्टेंट पैटर्न. So the highest resistant observed against cephalosporin, fluoroquinolones, and the reduced susceptibility seen in Pseudomonas aeruginosa, Klebsiella pneumoniae. तो ये भी जो सबसे ज्यादा हाईएस्ट रेसिस्टेंट केस जो है वो भी सबसे मतलब मोर देन 60 के एज ग्रुप में पाए गए हैं, एंड लोवेस्ट रेसिस्टेंट जो है वो 0 to 20 एज ग्रुप में पाए गए हैं. और सबसे ज्यादा जो सेंसिटिव एंटीबायोटिक्स हैं वो ऑब्जर्व विद कोलेस्टीन, टीजीसाइक्लिन, लाइनेजोलिड एंड अमेकासिन. So next, this graph shows the highest sensitivity and the highest resistant antibiotics. At the age of 21 to 40 years, highest sensitivity and good response to the linezolid and colistin. And at the age of the 41 to 60 years, moderate sensitivity and mixed organism pattern. And at the age of more than 60, lowest sensitivity and high resistance. सो इन डिस्कशन इसके अंदर मुझे सबसे ज़्यादा जो इन्फेक्शन है वो मेल पेशेंट में पाया गया है, एंड उसके बाद देन फीमेल पेशेंट में. लाइक सबसे ज़्यादा जो इन्फेक्शन है वो एज के अकॉर्डिंग अगर हम बात करें तो मोर देन 61 ईयर्स के एज में सबसे ज़्यादा इन्फेक्शन पाया गया है, एंड मोस्ट ऑफ द ग्राम नेगेटिव बैक्टीरिया है वो ज़्यादा अफेक्ट कर रहे हैं मेल पेशेंट को. एंड जो एंटीबायोटिक्स है, एंटीबायोटिक्स के केस में मुझे सबसे ज़्यादा रेसिस्टेंसी जो है वो हाईएस्ट एज ग्रुप में पाई गई है, एंड वो है एसिफेलोस्पोरिन, फ्लोरोक्विनोलोन्स, अमोक्सिसिलिन, और एमडीआर भी मल्टी ड्रग रेजिस्टेंस भी हायर एज ग्रुप में पाया गया है. सो कंक्लूजन इज दैट कि this paper shows that the gram-negative bacteria were the leading cause of respiratory tract infection, with Pseudomonas, Escherichia coli, Klebsiella being the most prevalent isolate. So, all in all, it can be concluded that the important role of constant monitoring, rational administration of antibiotics, and strict infection control measure is necessary in both high-risk elderly group, because it will help to manage respiratory infection and reduce the propagation of the resistant organism.
Okay, yes, Manisha, thank you. So
Okay.
you told me that the prevalence of the gram-negative bacteria is higher in the elderly patients.
Yes, sir.
What is the main reason behind that? Why the lung function get more infection from this particular gram-negative bacteria, not gram-positive bacteria?
सर ग्राम नेगेटिव बैक्टीरिया इज हाईली वर्नरेबल, मतलब बहुत ज़्यादा इन्फेक्शन हमारे गट फ्लोरा में भी होता है, और बहुत मतलब जैसे हॉस्पिटल एक्वायर्ड भी है ये काफ़ी ज़्यादा, लाइक सीडोमोनास ओरिजिनोसा जो कि क्रॉस कंटामिनेशन से मिलता है. तो इसलिए ग्राम नेगेटिव बैक्टीरिया ज़्यादा वर्नरेबल है एज कम्पेयर टू ग्राम पॉजिटिव.
Give me one example of gram-positive bacteria which is quite causing infection or disease state in human body.
स्टेफाइलोकोकस ऑरियस कॉज करता है इन्फेक्शन.
What type of
स्किन
what type
इन्फेक्शन.
of skin infection? It is same as the eczema.
यस सर.
eczema and the skin infection caused by this gram-positive bacteria, that is Staphylococcus, is the same clinical sign and symptoms.
No sir, sign and symptoms तो different है.
So then why you are making this statement that it is the same condition that is being caused?
इन्फेक्शन कोज करता है सर, ये स्किन इन्फेक्शन भी कोज करता है और मतलब लाइक हमारे रेस्पिरेटरी भी निमोनिया भी कोज करता है ये.
Okay, so what is your main focus in this entire study? Uh, at what point you want to make the highlighting that your study, your presentation is being focusing on this particular area, and it is dealing with this particular problem in the respiratory tract infection, which is quite common in elderly patient or geriatric patients. So what do you want to looking for this? What is the clinical
सर
implication of the study?
सर
Yes.
इसके अंदर अपने एज वाइज डिस्ट्रीब्यूशन कराए कि ज़्यादा से ज़्यादा मतलब जो मोस्ट हाईली एजेड पेशेंट होते हैं उनके अंदर अभी ज़्यादा इन्फेक्शन मिला है, तो इसके अंदर ये और एंटी माइक्रोबियल रेसिस्टेंसी भी उन्हीं के अंदर बहुत ज़्यादा मिली है, तो ये एक लैब डेटा है कि हम इसको मतलब आगे कंक्लूड कर सकते हैं कि हम कैसे इसे कंट्रोल करें अगर ये चीज़ हो रही है तो. और ये एक जैसे लाइक दिल्ली एनसीआर का रीजन सिलेक्ट करके इसको लिया गया है.
कौन सी लैब का डाटा आपने कोरिलेट किया है यहाँ पे? फ्रॉम विच लैब यू
सर डायग्नोस्टिक लैब है द दिल्ली एनसीआर में.
it is NABH aggregated?
यस सर, यस सर.
From which hospital it is has the collaborations?
सर डिफरेंट
Diagnostic.
डिफरेंट हॉस्पिटल्स हैं, डायग्नोस्टिक में काफी डिफरेंट डिफरेंट हॉस्पिटल से कोलैब.
See, Manisha, when you are doing any study on the PG level or on further as a research scholar, you always have to go with some authentic literature. Okay, and that should be proved. Otherwise, it make no sense to make all inclusion of all data in your study which is not yet proved. Okay, the importance of your study is to make the further platform for the further research. So there's a lot of thing you have to work on that area, and you should have some evidence based research study. Otherwise, there's no use. Okay,
ओके सर.
thank you, thank you.
ओके सर. ओके.
Hello.
Yes, participants, we will meet at 1:40 with new link. So you join new link. We will provide it in new group. Okay. We will provide and share in the group IC 2026. कर कर दो ओवर.
Over. कर दो.
सर इसमें ना फिल कर दो, हमें दूसरे में जाना. ठीक है, सो फिल एंड कर दो.